Ladies and gentlemen, it's a great pleasure and privilege for me to welcome you to the 2020 Nobel Lectures in Physiology or Medicine. My name is Ole Petter Ottersen, and I'm the president of Karolinska Institute. During the Nobel Week, we celebrate science. We rejoice at the unique contributions of some exceptional individuals whose discoveries, in the words of Alfred Nobel, have conferred the greatest benefits to humankind. Research is a way of quenching our thirst for knowledge, testing our own limits, and showcasing our creativity and competitive spirits. Within physiology and medicine, the ultimate goal is to achieve scientific breakthroughs that significantly deepen our understanding of human health and disease. The discoveries we celebrate this year are stellar examples of such breakthroughs. Today, it's an honor for me to extend my warmest congratulations on behalf of Karolinska Institute to Harvey J. Michael Houghton, and Charles M. Rice, who are receiving the 2020 Nobel Prize in Physiology or Medicine for the discovery of Hepatitis C virus. Without further ado, I now turn the floor over to Maria Masucci, Professor of Virology and member of the Nobel Assembly. Maria Masucci will introduce this year's laureates. Maria, the floor is yours.
Dear Nobel laureates, President of the Karolinska Institute, colleagues, ladies and gentlemen. The Nobel Assembly at Karolinska Institute has awarded the 2020 Nobel Prize in Physiology and Medicine jointly to Harvey J, Michael Houghton, and Charles Rice for the discovery of hepatitis C virus. Hepatitis, or inflammation of the liver, is a disease characterized by fatigue, loss of appetite, vomiting, and in many cases, a typical yellow discoloration of the skin and the white of the eyes, called jaundice. It is an old human disease. It was already known by the Babylonian, is mentioned in the Old Testament, and was accurately described by Hippocrates more than 2,000 years ago. However, only in the beginning of the last century, doctors understood that the disease affects the liver, that the vast majority of cases are due to infection, and that the agent or agents could pass through filters that captured bacteria, suggesting that they may belong to a recently discovered group of pathogens known as viruses. Today, we know that 5 different viruses may cause clinically distinct forms of hepatitis. The hunt for these viruses is one of the most fascinating scientific sagas of our time, and this afternoon we are going to hear one of the most thrilling chapters, the discovery of hepatitis C virus, or HCV as we call it. This was a momentous discovery. HCV turned out to be a very tricky and dreadful virus. It infects through contact with even small amount of blood, such as after unsafe needle injection, transfusion of contaminated blood or blood products, and other medical and non-medical practices that cause bleeding. But what makes HCV particularly frightening is that while a new infection is usually asymptomatic, more than 70% of the infected individual develop a chronic infection that in the course of years or decades will cause life-threatening liver failure and cancer. During this long asymptomatic period, the infected person continuously produces virus and can therefore accidentally transmit the disease. The WHO estimates that more than 70 million people around the world are chronically infected with HIV, and around 400,000 people die every year of HCV-related diseases, mainly liver cirrhosis and liver cancer. This makes HCV infection a global health burden comparable to malaria, tuberculosis, and HIV. The discoveries of this year's Nobel laureates have recognized a clinically distinct form of hepatitis, uncovered a new virus and produced reliable diagnostic tools, and have allowed the construction of an infectious virus clone, which finally proved that the virus alone caused the disease and opened the way for the development of new, highly effective antiviral drugs. It is a story of how scientific curiosity, careful and methodical clinical observation, technical ingenuity and creativity, resilience, and most certainly a bit of luck, have led to a discovery that has already improved the life of millions of people and may allow in a foreseeable future the eradication of a life-threatening viral infection. Without further ado, I would like now to introduce the first of today's speakers. Dr. Harvey J. Alter was born in New York in 1935. He received his medical degree from the University of Rochester in 1960. After residence at the Strong Memorial Hospital, he moved to the clinical center of the National Institutes of Health in Bethesda, where he worked with Barack Bloomberg on the Australian antigen, which became a major factor in the discovery of hepatitis B virus. Between 1966 and 1969, he worked at Georgetown University Hospital in Washington, D.C., where he also served as a director of the hematology department and was a strong proponent of the screening of blood donors for HBV infection. Dr. Alter returned to NIH in 1969 and joined the Department of Transfusion Medicine, where he performed the research for which he is now awarded the Nobel Prize. He remains to these days at NIH as Chief of Infectious Disease Section and Associate Director of Research in the Department of Transfusion Medicine. He continues to work on HCV, helping younger colleagues to uncover the last secrets of this fascinating virus.
As a clinical scientist, I'm an unlikely candidate to be a Nobel laureate. For that reason, I'm going to speak rapidly so that the Nobel Committee does not have time to reverse their decision. In truth, I'm deeply honored and humbled by the magnitude of this award. And I'm also gratified that the Nobel Committee recognized the power of observation and the power of non-directed research, where the end cannot even be envisioned at the beginning, and where time is allowed for this work to progress to its natural fruition. I want to thank NIH for giving me that time and the support that went with that time. Truthfully, I don't believe my part of this work could have happened anywhere else. I'm thankful also to the co-recipients of this prize, Michael Houghton and Charlie Rice, who brought these clinical observations to the next level in first cloning the hepatitis C virus and then in developing a full-length infectious clone that was critical to drug development. Mine is a long story spanning 4 decades that in the end saw the eradication of post-transfusion hepatitis and the development of new therapies, astounding therapeutics that can now cure virtually 100% of patients chronically infected with hepatitis C. And these findings portend that HCV could potentially be eradicated over time, even in the absence of a vaccine. In the time of COVID, this is a picture of intimate contact. Because of COVID, I'm giving this talk in the clinical center of NIH rather than in Stockholm. I'm saddened and disappointed that I can't be in Stockholm. Perhaps this was meant to be, because this is the room where it happened. One of the congratulatory cards I received had this on its front piece: "There is no elevator to success. You have to take the stairs." I have spent a lot of time on the stairs, and I found that research stairs, like every other stair, go down as well as up. Today I want to take you step by step through the early stages of HCV discovery as they occurred at the NIH, through my own work and that of many collaborators who I will acknowledge as I go through this talk. The hepatitis story actually began 2,000 years ago, when Hippocrates observed patients who were turning yellow, that he termed icterus. And then later he observed people with hardening of the liver, that he designated as chiros. He thought that these conditions were due to an imbalance of the four humors, in this case an excess of yellow bile. While this hypothesis did not hold up to subsequent peer review, it emphasized Hippocrates' great powers of observation and made him the father of medicine. In truth, Hippocrates was making a profound statement. That is, that disease was not due to divine punishment, but rather to some external force that perturbed the delicate balance of the body's systems. He did not know that these external forces were infectious diseases, or that a virus could cause hardening of the liver, but he made the critical distinction between divine intervention and early and earthly occurrences. Nothing much happened over the next 2,000 years, except that in every decade there were wars somewhere in the world. And in every war, soldiers came down with icterus, the acute symptoms of hepatitis. Thus, this was called campaign jaundice or vaccine-induced jaundice. And ultimately, McCollum and Havens and others distinguished two forms of hepatitis. One that was of short incubation and acute and led to recovery and was due to transmission by food and water. And the other of long incubation, the first called hepatitis A, the other of long incubation that was due to parenteral exposures and that could lead to chronic liver disease. That was designated hepatitis B. While these clinical and epidemiologic observations were made, nothing evolved in the way of specific visualization of the virus or of testing for the virus. And the breakthrough came in the early 1960s, here at NIH, when I was working in the laboratory with Dr. Baruch Blumberg. We were investigating polymorphisms in human beta lipoproteins. These typically, the precipitin lines that develop between a multiply transfused patient and some other normal subject, typically took up a blue stain for the lipoprotein. One day I observed a line which did not take up the blue stain, but stained red for its protein content. This line, because it was found in the serum of an Australian Aborigine, was tentatively called the Australia antigen. The ultimately linking of that antigen to the hepatitis B virus was a fascinating story that evolved over about 6 years and was attributed primarily to Dr. Blumberg and his group, who was then in Philadelphia. And for this, Dr. Blumberg won the Nobel Prize. And this fascinating story is chronicled in the Nobel Lectures of 1976. The relevance to my talk today is that this Australia antigen, subsequently called the hepatitis B surface antigen, provided the first serum marker for any hepatitis virus and laid the foundation for the events that I'll describe. At NIH in the late 1960s, a prospective study was established by Paul Schmidt, Paul Holland, and Bob Purcell that I took over in 1970. In this study, we used open heart surgery patients because they were heavily transfused and because they usually did not have confounding diseases or underlying immunodeficiency. We followed these patients every 1 to 2 weeks for the first 3 months after transfusion, and then monthly for an additional 3 months. And we saved the donor samples where we could. The only thing we could do to determine hepatitis was to measure liver enzyme elevations, particularly the ALT and AST. But later, because we stored these samples, we could go back with advanced technologies and PCR. The first thing we found, and quite astonishingly, that when you were following patients prospectively, that about 30% of these patients were developing post-transfusion hepatitis. Now, most of these cases were anicteric, but because we were prospectively following them, we could detect these enzyme elevations. The next thing that occurred in examining why this high rate of hepatitis, a study done by John Walsh, showed that it was dependent on the source of the donor blood. That if one received even 1 unit of paid donor blood, so-called commercial blood, you had a 51% chance of developing hepatitis, whereas if you received only voluntary blood, you had only a 7% chance. Thus, in 1970, we went, we went to an all volunteer donor system, and we introduced the first test for hepatitis B surface antigen. This caused a precipitous drop in post-transfusion hepatitis incidence from 30% down to about 10%. It was a dramatic change, even though the amount of blood given, shown in the little squares, was not different. It was just the source of the blood. The next events were that in 1973, Abbott Laboratories developed a much more sensitive test for surface antigen. We went back into our repository and tested all these samples, and somewhat to our surprise, we found that only about 25% of cases were due to the hepatitis B virus, and that there was some other non-B entity. Then, in 1975, Feinstone and Kapickian and Purcell, shown from your right to your left, discovered the hepatitis A virus using immune electron microscopy. Since Dr. Purcell and Feinstone were our key collaborators, we immediately sent samples to them for hepatitis A testing and found that not a single one of these non-B cases was due to hepatitis A. It was then in our brilliant bit of deductive reasoning that if we said that if these cases were not due to hepatitis A or hepatitis B, we would call it the non-A, non-B hepatitis agent. We deferred in calling it hepatitis C at that time because we hadn't yet proven that it was a transmissible agent, and if so, how many agents might be involved. Thus, we came up with this term non-A, non-B. The next step in this progression was to try to transmit this infection to the only available model, the chimpanzee. Chimpanzees did not get sick from hepatitis, but they did get ALT elevations. And here you can see our initial group lining up to sign their informed consent for these transmission studies. And what was critical was that we were able to transmit hepatitis from patients who had acute non-A, non-B hepatitis, patients who had chronic non-A, non-B hepatitis, and also importantly from asymptomatic donors who had been implicated in hepatitis transmission. In the next phase, a critical patient came to my attention. That is Mr. Hutchinson, who is now deceased and who did allow me to use his name. Mr. Hutchinson was an inveterate trailblazer. And one day, while blazing a trail. He suddenly collapsed and was virtually dead when his wife, who accompanied him, was able to give him CPR and revive him. And eventually he came to NIH. And he became a key participant, not only for my research, but also for research around the world. This is the course of Mr. Hutchinson's acute hepatitis, shown on your left, where in blue are the ALT elevations. And you can see that about 6 weeks after transfusion, where he received 17 units of blood, his ALT levels began to rise precipitously, reaching a peak of 2,112, where the normal value is 40. Then the ALT declined quite rapidly, and Mr. Hutchinson went into an asymptomatic carrier state for the next 30 years. He had no progressive disease and had liver biopsies that also showed no progression. The interesting point is that we were able to go back to his stored samples and able to obtain an apheresis unit at point A on the ALT curve, at a point when the virus would be presumed to be at its highest levels. And as you see at point A, he had 3 × 10⁷ RNA molecular units, and at that point, the sample was inoculated to a chimp, shown on the top right panel, and caused hepatitis in the chimp. And Dr. Purcell titered that material in the chimp model and showed that it had a titer of 1 × 10⁶.5 chimp infectious doses, almost identical to the molecular titer of 3 × 10⁷ genome copies. You see then that the HCV RNA level declined quite rapidly, coincident or just before the fall in the ALT level. At point B, at the peak of the ALT elevation, we inoculated a second chimp in the middle panel on your right. That chimp did not develop hepatitis. We think he did not develop hepatitis because the HCV RNA level was on a rapid decline at that time, and because, shown in the yellow bar, antibody had appeared by that point and was complexing the virus and making it less infectious. On the bottom panel on the right are differential sedimentation studies that showed indeed that the particles were heavier after antibody appeared, indicating the immune complexing. So now, at this point, we had 2 things. We had a titered infectious inoculum, and we had the chimpanzee model. Steve Feinstein then did this important experiment where he did a chloroform extraction of the Hutchinson material and shown that it was no longer infectious in the chimpanzee, whereas a sham extraction shown on the top was infectious in the chimp. This indicated that the virus, presumed virus, had lipid in its envelope and put it into a different class of viruses that we'll talk about. Next, in Dr. Purcell's lab, performed filtration studies where the filtrates through various size filter papers were inoculated into a chimpanzee to see where transmission was blocked. And it was blocked at about 30 nanometers. With these 2 experiments, we're able to say that the agent was lipid enveloped and that it was relatively small in size, somewhere between 30 and 60 nanometers. This narrowed the possibilities for what this agent may be to the alpha and flavi RNA viruses, so-called toga viruses at that time, to some variant of the hepatitis B virus or the Delta virus, but we had a lot of information that it was not related to hepatitis B. So it's getting down to being either a new class of viral agents or one of these small RNA alpha or flaviviruses. It was Dan Bradley at the CDC, who was doing work in parallel, who said that this agent was most consistent with being a flavivirus, as it ultimately turned out to be. We then went on to try to characterize this non-A, non-B agent, but it was a very, very slow process in developing a test. In the interim, we looked at the severity of the disease because there's questions of whether this was really a significant entity or just asymptomatic ALT elevations. So we then studied our patients with liver biopsies in collaboration with the NIDDK liver unit, and showed that our initial biopsy of 39 patients, that most had mild to moderate disease, but that 13% was then termed severe chronic active hepatitis, and 10% already had cirrhosis. We then did follow-up biopsies on those who had the chronic active hepatitis, and it showed that over time, in 20 repeat biopsies, that 30% actually appeared to improve somewhat. 45% seemed to have a stable histologic lesion, but 25% had progressive disease where they too developed cirrhosis. So in the end, we had eight patients out of 39, or 20%, who developed cirrhosis, an incidence that has held up over time. Not only that, but 3 of these patients died of their liver failure. 3 others had very severe liver disease when they died of the intercurrent cardiac illness. So we now knew we had a common and potentially fatal disease, and then people's attention focused on non-A, non-B. Getting back to the transfusion story, by 1970, the rates had come down because the amount of blood transfused had come down by about 2/3. And the rates settled in between 5 and 6%. We tried different interventions. We tried ALT testing of blood donors in 1981, but that did not seem to have an apparent effect. In 1985, HIV came along and an HIV test, and we screened our donors for HIV, thinking that might be helpful for hepatitis as a surrogate marker, but it was not, because the routes of transmission for HIV and HCV were different. We then did retrospective studies and showed that anti-core testing, that is, testing for hepatitis B anti-core antibody, predicted a 30% reduction in post-transfusion hepatitis. Excuse me. So in 1987, we introduced anti-core testing of donors, and it did have the expected effect, lowering the rates by 25% to about 4.1%. But we still, in all that time, could not develop a specific non-A, non-B assay. It was that point that in my frustration, I wrote this poem called "I Can't See the Forest for the HBAGs." I said, I think that I shall never see this virus called non-A, non-B, a virus I cannot deliver, and yet I know it's in the liver. A virus that we often blame, but which exists alone by name. No antigen or DNA, no little test to mark its way. A virus which in our confusion has forced us into mass collusion to make exist just by exclusion. But is it real or an illusion? Oh, great liver in the sky, show us where and tell us why. Send us thoughts that will inspire us. Let us see this elusive virus. If we don't publish soon, they're going to fire us. Well, I think this poem directly influenced Michael Houghton and his group at Chiron, because shortly thereafter, they cloned the non-A, non-B agent using early emerging molecular biology. Michael Houghton will go through this in his talk, but just rapidly, they pelleted known infectious samples, reverse transcribed the RNA, used the phage GT11 expression vector, and found with immunoscreening that a single clone turned positive out of what I understand were 6 million clones that were tested. And ultimately, they could subclone this, derive a small sequence which could then be expanded and develop an antibody assay that proved to be critical. This is a picture of Michael Houghton on the right at the time he won the Lasker Award, which I was happy to share with him, and a picture of Dan Bradley, the CDC, who was instrumental in the chiron discoveries. Shown here is a picture of me when I heard that chiron had cloned what I considered to be my agent. Not really my picture, but at that time I wrote a second poem that I said, "There's no sense chiron over spilt milk." It was a long poem, and I'm just going to read one paragraph. It was in Dr. Houghton's antiviral prescription to make clones by reverse transcription, while chiron's finance department had another conniption. But Michael said this was not a rehearsal. The RNA had to be changed by a transcription reversal. Like Jack the Ripper at his most vindictive, he chopped up the codon with an enzyme restrictive. Could these tiny little pieces be of an antigen predictive? Corporate executives prayed that it would, on their knees benedictive, showing that science and religion are not contradictive. Chiron then, when they had developed this antibody assay, came back to test, to request a panel that I had developed. It was a small panel, but everything was in it in duplicate, and the duplicates were placed in random positions. And 19 other laboratories who thought they had a non-A, non-B test were unable to break the code of this panel. But Chiron did. They broke it perfectly. They detected a positive antibody test in 3 persons with chronic non-A, non-B hepatitis in 6 duplicate samples. They detected it in 2 implicated donors in 4 random duplicate samples. They did not detect it in the acute post-transfusion hepatitis because the patients had not yet developed antibody, but later these samples were also positive. And very importantly, they did not detect the antigen in 7 negative controls, persons who had been blood donors on more than 10 occasions and never implicated in hepatitis transmission. They found a negative result in these 14 samples from 7 donors. So they had a perfect score. I then looked at 15 of our best non-A, non-B cases, and all 15 were negative for this antibody pre-transfusion and became positive post-transfusion. Then looked at the donor samples from 25 such patients and found a positive donor in 80% by a first-generation assay and 88% by a second-generation assay. So we could predict that if we use this test to screen donors, that we might prevent near 90% of post-transfusion hepatitis. And indeed, that was the case. In 1990, when the first-generation test was introduced, the rates went down to 1.1%, and after a second-generation test in 1992, the rates fell to virtual 0 by 1997. It can be retrospectively estimated that in the US, transfusions may have transmitted 4.8 million HCV infections in the 2 decades from 1970 to 1990. Conversely, HCV testing introduced in 1990 probably prevented 2.4 million additional transmissions in the next 2 decades. This is the same slide I just showed you, just with the addition of this arrow. This is not only the story of post-transfusion hepatitis, this is the story of my life. And you can see that both post-transfusion hepatitis and my life are on this rapid decline. However, it appears that the virus has gone away from transfusion, and I'm still up here talking about it. So that's encouraging. On a global scale, there are supposedly 170 million people who are chronically infected with HCV, about 5 million of those in the US. The critical point is that 75 to 85% of patients who get infected with HCV become persistent carriers. So we focused for a long time to see why does this virus persist so readily. This is a study we performed with Patricia Farsi. She was the main investigator, where she cloned 105, made 105 clones from the Hutchinson sample. 57% of those clones were identical, but at the same time, there were 19 other clones, 19 other variants already present in that single sample from this patient. Thus, if you developed even a very strong neutralizing antibody against a dominant strain, any one of these other strains could have become dominant and make the infection persist. This has made it very difficult to develop a hepatitis C vaccine. To show you how this works, we looked at early samples from the time of transfusion until the peak of the ALT elevation, doing 10 clones at each time point. At week 3, Patricia showed that there were 3 predominant variants, which are arbitrarily labeled A, B, and C. By week 8, D was now the predominant strain, but there was also E, F, and G. At that point, antibody appeared, and the variation increased. So now at week 13, you could see that A was still present, but no longer dominant. Now it was HIJK elemental P. And by week 18, 16, the alphabet went up to U. So you can see this progressive mutability and variation in the viral genome, each of one capable of carrying on the infection. The other problem with hepatitis C is that the high antigenic load arising very early in the infection can blunt the T-cell responses. On this slide, which is not from our laboratory, by Wertheimer, shows vividly that patients who have recovered from hepatitis C have good CD4 and CD8 responses. So the T cells are not destroyed, but during infection, they are blunted. So during chronic infection, CD4 and CD8 specific T cells are not detectable by this ELISpot assay and are no different than the normal population. Again, the blunting of the T cell response is critical in the persistence of the virus. In another study of T cells done by Von Hahn and Jane McKeating's lab, where we did collaborate, if you look only on the right, you'll see that the wild-type virus, the Hutchinson sample from 1977, had this short sequence in the E2 region of the virus, whereas a mutation in 2003 showed 2 nucleotide changes and 2 amino acid changes, and thus the antibody, which is highly effective against the wild-type strain from 77, no longer neutralized the mutant strain in 2003. From a clinical standpoint, these open-heart surgery patients showed us that there were 3 major forms of post-transfusion hepatitis C. The majority of patients had this very stable or slowly progressive disease, where though you could not distinguish this early in infection, they went on to become persistent carriers of the virus, but their liver histology remained relatively dormant, and they could live for 4 or 5 decades, maybe their entire life, without consequences. But 20 to 30% had severe and progressive disease where they developed cirrhosis in 15 to 40 years. A small group had cirrhosis more rapidly, and I think this group had confounding illnesses, or particularly alcoholism or NASH. To look at what the difference is between the slow and the rapid progressors, Patricia Farsi, again, my primary collaborator in recent decades, did this study and showed very importantly that slow progressors, as shown in yellow, had a rise and then a rapid decline in the HCV RNA level where it almost disappeared by week 20, but didn't quite disappear when a mutant evolved and the levels of the virus went back up again, and led to the chronic state, whereas those with rapid progression never achieved this initial containment of the virus. Also, the rapid regressors were found to have high levels of MCP1, monocyte chemotactic protein. This is a profibrogenic chemokine that both attracts stellate cells and induces the stellate cells to produce fibrotic tissue. In contrast, the slow progressors have increased levels of interferon gamma and MIP1 beta, which are favorable cytokines that would help in recovery. In other studies with Kentaro Matsura from Nagoya, Japan, we showed that did microRNA analyses and showed that one microRNA, LET7, was particularly differentiated between those who had progressive fibrosis and those who had mild disease. And found, too, that the importance of this is that LET7 is a suppressor of TGF-beta, and TGF-beta is a major stimulant of hepatic stellate cells and collagen production. In this particular study shown on the left, we looked at serial biopsies of patients, looked at the LET7 levels of patients, and showed that their levels, which were initially lower than those with slowly progressive disease, became progressively lower as the fibrosis increased. So we think this may be a marker of fibrotic progression. Another interesting study we did with Japanese investigators, this was Yasuhito Tanaka and Dr. Mizukami, again in Nagoya at that time, looking at a molecular clock. Now, a molecular clock is a mathematical analysis that allows you to determine when a particular variant or genotype of the virus diverged from some common ancestor, and then when it spread rapidly within a given population. You can see that in Japan, genotype 1b diverged around 1880, and then was stable until the 1930s and 1940s when it rose dramatically. What was going on in Japan at that time was Japan was at war with China, and then at war with the US and the rest of the world. We learn now, in retrospect, that the Japanese soldiers were shooting up with amphetamines and sharing needles, and this was the cause of the rapid rise in HCV in that population. In the US, on the other hand, the virus diverged, the genotype 1A virus diverged around 1910, and then didn't rise until the 60s, 70s, and 80s, when we had our own drug epidemic with needle sharing, which is now still unabated. The key point of this slide is that there's about a 30-year to 40-year differential between the spread times in Japan and in the US. And this is critical because Japan has an 8 to 10-fold increase in hepatocellular carcinoma compared to the US. And the Japanese have always said that you will have the same rates. It'll just take some more time for that to develop. And in fact, they were correct. On this slide is shown a study by El Sarag, where if you look at the incidence of hepatocellular carcinoma in the late 70s compared to the late 90s, you can see that there's a 3-fold increase. And that was all due to hepatitis C, as shown on the right, not to HBV or not to any other known cause. It was assumed at that time that unless there were some changes in the standard of care, that the total number of patients with advanced liver disease and hepatocellular carcinoma by 2029 are projected to be greater than 4-fold higher than they were in 2009. Fortunately, there has been a major change in the standard of care. And treatments for HCV have evolved to remarkable levels. I won't go through these various, I won't go through these various iterations of treatment, but since 2011, where direct-acting antivirals, HCV-specific inhibitors were developed and have had dramatic effect on the treatment of these patients. So now 95 to 98% of patients with hepatitis C can be cured, in some studies 100%. So how you view hepatitis C depends on whether you see the glass as half full or half empty. If you see it as half full, you could say that with cure rates now approaching 100%, from this time forward, once HCV infection is identified, no one should develop cirrhosis or die from hepatitis C sequelae. But there is a half-empty side of that glass. That is, it's estimated that only 50% of HCV infections have been identified. At least that number remain unidentified. And this is much higher in the developing world. So enhanced population screening is priority one in the eradication of HCV. The second downside is that even among known carriers, only a minority have been treated with direct-acting antivirals, though this proportion is increasing. Third aspect is the drugs cost a lot, and that drug cost is now the main impediment to preventing the cure of millions of patients. The cure is no longer constrained by science, but is a matter of dollars. So obviously, to eradicate HCV, the main hope would be to develop a vaccine. That would be the holy grail of eradication. But thus far, it's been very difficult to develop a vaccine for the reasons I've indicated, although work continues. But I want to just discuss, as the last point, what are the possibility that HBV or HCV could be eradicated, even in the absence of a vaccine. Now, for hepatitis B, we have a very effective vaccine, which is being widely spread throughout the world. We also have nucleotide and nucleoside inhibitors, which can lower HBV viral loads to undetectable and thus decrease transmission. For HCV, we have these direct-acting antivirals with greater than 90% cure rates, but we don't have the vaccine. The goal then of global eradication depends on some intermediate step. I've labeled this as, and then a miracle occurs. But what are the elements of that miracle? First, we need universal vaccination for HBV. This is in progress. The World Health Organization and others are doing very well in getting people vaccinated around the world, but it's not up to the 90% level that we need. For HCV, we need to detect almost all HCV carriers through massive global screening. This is a very difficult task, but it's achievable. And then we need to have the global delivery of direct-acting antivirals with a greater than 90% penetration within each population. I can envision that with a rapid test for HCV RNA, a patient could be tested and, if positive, immediately be given 3 months' worth of pills that they could take home and administer without additional medical care, because there are virtually no side effects from these drugs, and therefore effect a cure. So the cure now depends on the political, corporate, philanthropic, and moral will to make it happen. It will take decades, but I think it could happen even without a vaccine. But it will not happen under my, with my help. I'm too far along in the course. It will take young investigators, and that's fitting. And I have developed a new philosophic perspective of life with increasing age, shown here in this slide, where age on the abscissa is plotted against give a damn on the ordinate. And this is me here at the end of this curve. There are so many great young investigators in this field now that all these goals will be achieved and can be achieved. And I want to just end with another poem, one that I did not write. Ralph Waldo Emerson beat me to it, but I think it is so fitting definition of success. He said, "Success, to laugh often and much, to win the respect of intelligent people, to earn the appreciation of honest critics, to appreciate beauty, to find the best in others, to leave the world a bit better, to know even one life has breathed easier because you have lived. That is to have succeeded." So I want to thank the Nobel Foundation and the Nobel Selection Committee for validating that I've met at least some of these Emerson criteria. Thank you very much.
Our next speaker will be Dr. Michael Houghton. Michael Houghton was born in the United Kingdom in 1949. He graduated in biology at the University of East Anglia in Norwich in 1973 and obtained a PhD in biochemistry at the King's College in London in 1977. He subsequently moved to the United States, where in 1982 he joined the California-based biotechnology firm Chiron Corporation. At Chiron, he worked on what was at the time known as non-A, non-B hepatitis, and for the first time succeeded in isolating novel viral sequences using exclusively molecular and immunological methods. Dr. Houghton left Chiron in 2007, and in 2009 joined the University of Alberta in Canada as professor of medical microbiology and immunology and director of the Li Ka-shing Applied Virology Institute. He is currently working on the development of an effective HCV vaccine.
Hello, my name is Michael Houghton. I work at the University of Alberta in Edmonton in Canada. And today I'm going to relate to you the work that my colleagues and I did to identify and isolate the hepatitis C virus in the 1980s. So the work I'm going to relate is mainly work that was performed between 1982 and 1989. And my work continues at the University of Alberta trying to develop a vaccine against hepatitis C. But today I'm going to focus on the work we did to identify it in the 1980s. So in 1982, I trained in England, and then I moved to a company called Chiron Corporation, set up by Dr. Rutter and Dr. Penhote, both of University of California. And after arriving in the fall of '82, I discussed various programs with Dr. Rutter and Dr. Diener and Dr. Valenzuela. And Dr. Diener introduced me to non-A, non-B hepatitis. And we realized that maybe some of the methods I had used in my work on interferon genes in England could be used to, could be applied to non-A, non-B hepatitis with a view to identifying the causative agent. So I was a molecular biologist, and looking at what we had available to apply to non-A, non-B, there wasn't much. There were infected human and chimpanzee materials available from different parts of the world, but polymerase chain reaction had not yet been discovered, and it wasn't until the late '80s that it started to be applied to labs on a routine basis. No antigen or antibody had been defined for non-A, non-B hepatitis, no cell culture system, no high throughput sequencing was available like it is today. And so, in other words, we didn't have a molecular handle. The work started off with a collaboration with Dr. Tatsuo Miyamura of the Japanese NIH. He came over with non-A, non-B liver samples from human cadavers, and he worked in my lab for almost a year. He also brought over non-A, non-B infectious blood from Japanese patients. we found that the quality of RNA that we could extract from the post-mortem liver samples was very poor due to degradation in vivo, excuse me. So we realized that we needed to work with a collaborator who had an animal model working. There were several groups in the world who had a chimpanzee model for non-A, non-B hepatitis. We approached Dr. Bradley at the CDC, who was in Phoenix, Arizona at the time. He was an exponent of the chimp model. I'm showing two examples of transmission of non-A, non-B hepatitis from humans to chimpanzees that in his lab. We asked him if he would be willing to collaborate and provide samples for our molecular biological approaches, and he agreed. The main program, the main analysis that we used at the time was a plus-minus hybridization approach. So basically, we cloned polyadenylated mRNA from infected livers. We converted it into cDNA first with reverse transcriptase, clone the cDNA into bacterial colonies, transfer the colonies to filter paper, and one can duplicate those filters, and you end up with a set of identical prints of the bacterial colony libraries. And we hybridized highly radioactive cDNA, either from infected liver RNA or from control uninfected liver RNA. And we're looking for signals that hybridize with the infected liver, but not with the control liver cDNA. So a laborious approach, one that we applied to many millions of clones. And at the end of that exercise, we had identified a lot of genes upregulated by non-A, non-B hepatitis infection, but none of them that we could consider to be viral in origin because they all hybridized to the chimpanzee and human genomic DNA. So they were all chromosomal derived. So we thought, well, we better have pedigreed samples, and we better have samples of higher titer if we were to stand a chance at identifying this elusive agent. So we did request to Dr. Bradley, and we co-funded his efforts to do this, to identify samples of higher infectivity titer. What Dr. Bradley did is to go back through his archives of chimpanzee samples from many chimps, and he found a chimpanzee, number 910. We're looking at the ALT profile over the course of years. ALT is an aminotransferase enzyme that is released by damaged hepatocytes as a result of the infection. And so what you're looking at is a very typical example of non-A, non-B in chimps and in humans, where you have fluctuating peaks of ALT and hepatitis. What Dr. Bradley did is to make pools from around the peaks of ALT over many years. He came up with a large plasma pool that he then titered in other chimpanzees. So by diluting and transmitting the diluent to chimpanzees looking for hepatitis, he was able to conclude that this plasma pool had an infectious titer of at least 1 million infectious doses per mil. Similarly, the liver from this animal, chimpanzee 910, had an infectivity titer of around 10 million infectious doses, chimp infectious doses per gram. So we now had some better materials, better pedigreed materials. We applied the hybridization approach that I mentioned to you earlier. Once again, found a lot of genes upregulated by non-A, non-B hepatitis, which were of interesting nature for sure. But once again, none that could be shown to be extrachromosomal and therefore unlikely to be of viral origin. During this time, my lab was applying lots of other methods to try and identify Hep C. There was a lot of literature suggesting it might be related to a known virus, for example, hepatitis B or A, yellow fever, or bovine viral diarrhea virus, a toga virus. So we used these known genomes as hybridization probes under low stringency hybridization conditions, but could never see a specific signal. And we know why now, because Hep C is a very unique virus. We did wonder in the mid-80s if Hep C was related to hepatitis delta virus. There was some evidence from the NIH suggesting that there were similarities, perhaps suggesting a sequence relationship. So my lab got involved with John Geron at the NIH, and we identified the genome of hepatitis D virus, otherwise known as Delta virus, for the first time. And this is an electron micrograph performed by Dr. Jim O of the genome. You can see that this is a circular piece of RNA. If you treat the RNA with formaldehyde at high temperature, like Jim did, you can actually see this circular piece of RNA around 1700 nucleotides in length. It's single-stranded, it's covalently closed, and it's a remarkable genome to occur in mammalian cells because it's highly related to plant viroids and virusides. So it's an unusual virus element to find in mammals, which was an interesting story. But the key question is, could we use it to identify Hep C? Unfortunately not. My colleague, Dr. Wiener, hybridized the whole genome of Delta, 2 extracts from Dr. Bradley's non-A non-B infected livers and plasmas. And the spots you see are due to positive controls that we included. That the key area on these plots is A4. That's where Amy had extracts of the high titer plasma pool from 910, and you can see nothing, even under low stringency conditions shown on the right. So once again, we could conclude that Hep C, whatever it was not related to hepatitis D. We were always pursuing other avenues. We had a lot of effort in trying to culture the non-A non-B agent in various cell lines. We now know that it's very difficult to do even today. Dr. Wakita in 2005 found a specific strain, one specific strain, that he was able to grow fairly well in tissue culture, in cell culture, but all the other strains are very difficult to grow. So this has been a very difficult virus to work on. We looked for reverse transcriptase activity in case it was a retrovirus. We could not see any evidence for RT activity. We looked for high molecular weight viral genomes, be they RNA or DNA, on electrophoretic gels. Couldn't see anything, either RNA or DNA. We also spent a lot of time trying to produce monoclonal antibodies that histochemically would bind to non-A non-B infected liver sections, not to control sections. We got antibodies to, once again, upregulated gene products, but nothing that we could decide was truly against the virus. About this time, Drs. Young and Davis at Stanford had developed a sophisticated cDNA expression system. Using a bacteriophage, lambda GT11, this was a highly efficient cloning vector, and each piece of nucleic acid that was inserted into this bacteriophage could be persuaded to express its encoded product into protein. And so one could easily produce very large proteomic libraries. And what Young and Davis showed, and some people before them actually, that you could use antibodies, characterized antibodies to specific proteins to fish out clones that way. And the process by Drs. Young and Davis applied to lambda GT11 was a very efficient one. So this was something we got interested in. Earlier in the program, I and others had considered this a very risky approach for a number of reasons. One is it didn't always work, even when a good antibody was available. No antibodies had been identified for non-A non-B hepatitis up to that point. And the persistence of non-A non-B suggested that there might be a weak immune response to this virus. I went next door to discuss antibody screening with my colleague and friend, Dr. George Kuo. He was not working on Hep C at that time, but he was a very skilled scientist in immunology and similar technology. And I explained to him how probably antibodies were limiting in non-A non-B. He suggested that maybe the reason why antibodies have not been identified so far was because of limiting antigen concentrations. Typically, people use liver sections of infected livers, as the target for antibody immunofluorescence assays. So George thought that it could be limiting antigen that was the problem, and he recommended that if we use cDNA expression, by definition, you could perhaps get high levels of antigen expressed via GT11. And with those high levels of antigen expressed, we might be able to identify antibodies for the first time, and of course, in so doing, identify viral clones. So this was a very persuasive argument from George. Around the same time, Dr. Bradley was discussing using the same approach, the expression approach, cDNA expression approach, with another group. And so he was also in favor of this and suggested we go forward trying it. He later supplied some of the chimp 910 plasma to the other group that he was working on, as well as to ourselves. Unfortunately, we didn't get the answer quickly using this approach. Started off making a GT11 library from polyadenylated RNA from 4 different infected chimpanzee livers from Dr. Bradley. Dr. Kuo looked at various methods of detection and decided that the use of I-125 labeled anti-human IG was able to achieve maximal sensitivity of detection. And then Dr. Chu screened this complex library using various convalescent sera from chimpanzees and patients. We all thought that using sera from individuals that had undergone acute hepatitis but then had resolved the hepatitis spontaneously without the use of antivirals, that would indicate perhaps that those individuals had the highest immune response, including antibody response. So we tended to use convalescent sera throughout this methodology. However, we obtained no viral clones, and then we switched to chimp 910 plasma pool, and once again, we could not identify any viral clones. So it was a frustrating time. At this point, we took a time out, so to speak, and we wanted to validate the whole cDNA expression system using hepatitis D virus. As I explained earlier, my lab had identified the hepatitis D genome, had identified antibodies to the virus, and so we had a supply of plasma with the virus in it, and we had a supply of patient sera with proven antibodies to the agent. So Kangsheng Wang and myself used HDV monkey plasma as a cloning source, excuse me, and we used HDV infected patient sera as a known source of antibodies, put the two together, and it worked very well. We got hundreds and hundreds of positive clones, and when we sequenced them, they were all hepatitis delta, virtually all hepatitis delta. So that was good news and reinforced our approach, but the delta monkey plasma infectivity titer was at least 5 logs higher than the non-A non-B chimp plasma 910. So that's 100,000-fold difference in infectious titer. Extrapolating the number of clones we got for delta to the number of clones we might get with delta, I came to the conclusion that, well, it's on the borderline, but we may be able to get this to work. So I returned to the non-A non-B plasma. I made a second cDNA lambda GT11 library from the plasma pool 910. This time we did do something different. Dr. Chu and myself, we chose to screen using a serum from a US patient who had abnormally high ALT levels. Normally, ALT levels in a chronic patient are in the 100 to 150 range. This patient, for some reason, had ALT levels in the 300s, 400s persistently. So it was an unusually strong hepatitis in this person. And we thought, well, maybe that indicates higher replication. Maybe this might be a better source of viral antibodies. And indeed, later on, after identifying Hep C, we did show that chronic patients have higher antibody titers than patients that have gone through an acute resolving phase of infection. But at this point, we were trying to think of anything different that we could try to identify this virus. And success at last. This happened around 6 years after we started in 1982. And I took the chimpanzee plasma pool, diluted it, ultracentrifuged such that the smallest known virus to man would be pelleted. I extracted total RNA and total DNA from that pellet, converted it into DNA using reverse transcriptase that we could clone into GT11, and then Quilem incubated serum from that patient with strong hepatitis with all the proteomics library from this plasma. And we were able to show that it truly was, I'm sorry, if I go back to this slide, we got some false positives, as you would expect with this method. We got one clone, 511. It was only 100 base pairs. And this library that I'm talking about was the only library made from plasma that actually had viral clones in it. Out of 6 or 7 libraries that I made, this was the only one that had hepatitis C clones in it. And out of all of those libraries, we only got one clone by this immunoscreening method. And as I said, it was 100 base pairs in length, which is very small. So we were able to get overlapping clones, overlapping 511. We were able to show that for the first time in all of our work, this clone was not derived from the chimpanzee or human genome shown on southern blots. We showed that it was derived from RNA from infected liver, but you could not detect its binding to control livers, shown here. The virus contained RNA. It was sensitive to ribonuclease, not to deoxyribonuclease, and it was single-stranded. If you made single-strand hybridization probes, only one of the strands hybridized, shown here. The other strand did not. This proved that it was actually positive-stranded in the sense that the RNA encoding 511 was the actual RNA in the virus, what we call a positive-stranded RNA virus. Also, the RNA was very large. This is polyadenylated RNA extracted from the chimp liver, run on a northern blot, and this top band here is around 10,000 nucleotides. So we were dealing with a large RNA genome, positive-stranded. We then asked Dr. Bradley to send us acute hepatitis samples from chimpanzees that he had infected with non-A, non-B, or with hepatitis B, or with hepatitis A. And of the 4 non-A, non-B animals, we saw seroconversion to 511 in all 4 non-A, non-B chimps. We did not see it in 7 animals infected with Hep B or Hep A. This was a very important finding, clearly showing that these antibodies to 511 developed only after infection with non-A, non-B. We then obtained additional proof for non-A, non-B etiology. We had 11 patients from Dr. Gary Gitnick, clinically diagnosed non-A, non-B cases. George developed a 511 immunoassay, radio immunoassay at this point, showed that 7 out of 11 were positive versus 0 out of 10 in controls. We were also getting more and more sequence from this viral RNA as we were walking the genome, as it's called, and we showed that it had a single large open reading frame. When we looked at the sequence very carefully, we could see very distant sequence identity with flaviviruses like dengue. It was very, very small distance homology, but nonetheless it was present enough to indicate this was a flavy-like agent. And so now, in the fall of '87, we believe that 511 was derived from a non-A, non-B hepatitis viral genome. The proof of the pudding is in the eating. Then we had to, and obviously very willing to collaborate with groups around the world. One of the first groups was the one at the lab of Dr. Harvey Alter, and he had made a panel of sera containing either proven infectious non-A, non-B sera, infectious for chimps, or also containing proven negative samples that did not transmit to chimpanzees. And when George used the radioimmunoassay to assay this panel, for the first time ever, this panel was correctly cracked, decoded. 6 of the 7 probe and infectious samples we could detect. As you can see, we've got counts in the many tens of thousands in all except for one sample, this one. So 6 of the 7 were scored positive. The pedigreed normal controls were all negative. This was background level in this assay, as were disease controls. So for the first time ever, we were able to break Harvey's code, which was very good news. We then looked at post-transfusion cases from him. He gave us 10 cases of post-transfusion non-A, non-B hepatitis. We were able to detect 511 in all 10. And he also provided us with the donors implicated in those post-transfusion cases. And in 9 out of 10 cases, we were able to identify at least one positive donor. We then went on and looked at other US non-A, non-B cases from Drs. Gitnick, Redeker, Dienstag, Stevens, and Tepmeyer. And of 24 samples, we scored 17 as positive, which was 71%, versus a background of 0.5% in healthy blood donors in the US. And finally, we scored 58% of Dr. Miriam Alter's sporadic non-A, non-B cases as positive in our assay. We then turned worldwide, and Drs. Benino and Colombo gave us 32 samples of chronic post-transfusion non-A, non-B hepatitis cases from Italy. 84% were positive. And then we got a panel of chronic post-transfusion non-A, non-B cases from Drs. Katayama and Miyamura from Japan. And we found 78% of those positive in our assay. Interestingly, Drs. Katayama and Miyamura also sent us 13 acute resolving cases of post-transfusion non-A, non-B hepatitis, and we detected only 2 of those, 2 out of 13, indicating 15%. And as I said earlier, it turns out acute resolving patients have less antibody titers than chronically infected cases. So our search for the virus ended in 1989. After 7 years with the publications in Science. We had back-to-back publications. The first one showing evidence that we really had identified a cDNA clone from blood-borne non-A, non-B hepatitis. I've mentioned all the names to you already, except Dr. Overby. He was a hepatitis expert who really formed the infectious disease diagnostic division at Abbott Laboratories, and he joined us in 1983 or 84, I can't remember exactly. But he was a great source of information about viral hepatitis in general. He was a great mentor and a great supporter of our work, so we are indebted to him. And then the second paper back-to-back was the one from Dr. George Kuo and Hui-Lim Chu, Harvey Alter, and all of our collaborators, and obviously Dr. Bradley, where we looked at different human samples from all over the world and showed that indeed Hep C was the major blood-borne non-A, non-B hepatitis virus. Well, excuse me, we walked the genome until we got to the ends, and it's about 10,000 nucleotides long, the viral RNA. It encodes for a lot of different proteins. It actually encodes for one single large polyprotein precursor that is cleaved through viral enzymes as well as host enzymes into a number of different proteins. And this was really, I suppose, the birth of the hepatitis C field. Now academics, industry, and public health could get involved in using this information of the genome to develop diagnostics. We had already got one with 511. 511, by the way, was derived from NS4 protein, a non-structural protein 4A. As we walked the genome, we could identify many other B-cell epitopes elsewhere in the viral proteins. So over time, we delivered more and more sensitive antibody diagnostics. The 5' untranslated region of the viral genome has a very precise secondary and tertiary structure to which ribozymes bind to initiate translation of the protein. It turns out my colleague Dr. Han showed that this is a highly conserved region of the genome, and therefore is exquisitely suitable for PCR assays and TMA assays to detect viral RNA in samples. So over time, we developed very good genome nucleic acid specific tests as well as the antibody tests. Many groups got involved in identifying the replicase encoded in NS5B. NS5A became a major antiviral target from scientists at Bristol Myers Squibb. This protein is involved in virus assembly and secretion, and it's very important. And scientists there developed very good antivirals against it. And many groups working together identified an inhibitor of the 5B polymerase, including Dr. Michael Sophia. In the NS3 region is a protease, a serine-like protease that has formed the basis for generation of specific drugs that have been used in humans very effectively. And then at the N-terminal region of this polyprotein, we see the two envelope proteins, the nucleocapsid protein. And this information has led us, and my group still working on it in the University of Alberta, to develop vaccines based on the recombinant envelope proteins, as well as T-cell epitopes throughout the non-structural region. So the genome has provided the platform for the development of diagnostics, therapeutics, and vaccine candidates. It remains only for me to say, once again, that the identification and isolation of this virus was a team effort. And I really would like to acknowledge the contributions of Dr. Chu, Dr. Kuo, Dr. Bradley, without whom I would not be talking to you today. Thanks again very much to the Nobel Committee for this honor and for honoring the hepatitis C virus field. We're very grateful. Thank you.
Our last speaker will be Dr. Charles Rice. Charles Rice was born in Sacramento, California in 1952. Following his early interests, he graduated in zoology at the University of California in 1974. But after a summer internship at the Marine Biology Institute in Woodsall, he became fascinated by basic research and moved to the California Institute of Technology, where he studied biochemistry and virology, earning a PhD in 1981. Dr. Rice remained at Caltech, working on various aspects of RNA virus biology until 1986. He then moved to Washington University School of Medicine in St. Louis, where he started his work on hepatitis C virus and made the discoveries for which he is now awarded the Nobel Prize. In 2001, Dr. Rice was recruited by the Rockefeller University in New York, where he continues to this day his groundbreaking research on HCV, biology and pathogenesis, as professor of virology and director of the Laboratory of Virology and Infectious Diseases.
To begin, I want to thank the Nobel Committee and organization for recognizing the hepatitis C story and allowing me and my esteemed colleagues and friends, Harvey Alter and Michael Houghton, to represent the many in this field who made this happen. I'm humbled and deeply honored to be awarded this prize, but as you will learn shortly, this really came about through a series of chance events. Let's begin with some history and context on how we came to work on hepatitis C. Even going back into the 17th and 19th centuries, there were outbreaks of jaundice. This actually traces back even to Greek and Roman times. Really sort of unexplained outbreaks. In the 1930s, it was noticed that vaccinees with yellow fever vaccination that had been stabilized with human serum often came down with hepatitis or jaundice. And then in the 1930s, later in the 1930s, there was recognition of a link between hepatitis in people and human serum. In the 40s, it was recognized that there were two types, infectious, which was probably the form that was a problem sort of during wars and outbreaks, versus serum, which I just mentioned. And then a huge breakthrough came in 1965 when Baruch Blumberg discovered that his Australia antigen was actually the surface antigen of hepatitis B virus. And this sort of led to the development of a vaccine for hepatitis B and other advances. So in 1973, in this picture, we see Steve Feinstein, Al Kapikian, and Bob Purcell using immunoelectron microscopy to sort of identify the hepatitis A virus particle. So this happened sort of in the early 70s, and then it became possible to sort of develop assays that could be used to screen blood products for both of these viruses. And it was noticed that by Harvey Alter and others that there were still significant cases of post-transfusion hepatitis. So they named this non-A, non-B hepatitis. So this was really a founding sort of breakthrough in the field. It occurred in about 10% of transfused patients. It often resulted in persistent hepatitis, and sort of later in the 70s it was shown that these individuals could go on to develop fibrosis and liver cirrhosis. Also at that time, several groups succeeded in transmitting this agent to chimpanzees. And that really sort of formed the sort of cornerstone of the field, and it was determined with these chimpanzee studies that this was likely to be a virus. It was filterable, and it was likely to be enveloped because it was inactivated by organic solvents. So this really catalyzed a great deal of effort among sort of many individuals to actually sort of identify this mystery virus, this agent. And that took almost 15 years from the time that it was noticed to be a problem until 1989. And this really, I think, was another sort of landmark in this effort. And that was led by Michael Houghton and his team at Chiron that had been making expression libraries from pooled chimp serum that was produced by Dan Bradley at the CDC. And they used this sort of expression screening approach to identify a single small clone that was immunoreactive with what they believed to be serum from a convalescent non-A, non-B hepatitis patient. So this was where we were in sort of 1989. And the thing that caught my attention about this is shown here. If we look at the abstract of the science paper from Mike and his colleagues, this was an RNA, a positive stranded RNA, and it seemed to be possibly related to the flaviviridae. And this was sort of based on these properties that I mentioned before, small, filterable, and inactivated by organic solvents, and also the sort of genome organization of this that was revealed by this sequence of approaching 10,000 nucleotides. So it turns out that in 1989, we were actually studying the prototype member of this family of viruses, yellow fever virus, which is a virus that many of you may know about. And I got a phone call on the lab phone from Steve Feinstein. So as I mentioned, Steve was involved in the sort of co-discovery of HAV. He'd also been involved in a number of the chimp studies on non-A, non-B hepatitis at the NIH with Bob Purcell. And he basically sort of called me up out of the blue on the phone and said, "Hey, Charlie, how about making a yellow fever 17D HCV chimera as a vaccine?" So Steve was then at the FDA and interested in making a vaccine. And at that time, we had been already busy working on yellow fever. The 17D vaccine strain had been developed by Max Tyler in the mid-30s. It's been in about 500 million people. It's one of the most safe and effective sort of human vaccines to date. We had sequenced this and reported that in 1985. We were also sequencing the virulent parental virus, the Acebe strain, and we had established an infectious clone for 17D, which basically, I'll talk more about this later, but that allows you to basically manipulate the genome as DNA and then sort of investigate the effects of those mutations on the virus life cycle. So this was the paper that was published in the New Biologist, a new journal at the time that shortly thereafter went extinct. And the last sentence of this, which I think maybe you can see here, said, this may allow yellow fever 17D to be used as a carrier for immunologically important epitopes from other disease agents. And this is what I think caught Steve's attention. So getting back to this, I think we got to talking about this possibility, but we really realized that we knew very little about the hepatitis C genome and how it was expressed. So we did know that at that time that this was a major problem, and with more than 100 million people that were chronically infected, that became possible sort of when diagnostic tests were developed shortly after the discovery of the virus. And as Harvey and others had shown in the 70s and 80s, this was an infection which often was chronic, usually asymptomatic, but that chronic infection in some individuals could lead to cirrhosis and severe liver disease. So this was really a problem of great magnitude. So that really sort of brought up the sort of goals post-1989. Clean up the blood supply. This was done sort of very quickly in the early 90s. The high-risk groups try and educate them, maybe develop a preventative vaccine. And then about successful treatment, one had to identify those infected and improve treatment. And then the hope at that point, although this was a bit of a pipe dream, was that we might be able to sort of eradicate this virus. So what do we do? I mean, the problem was that there were some challenges for hepatitis C. This virus had not been shown to replicate in any cell culture, despite sort of repeated attempts to see if you could amplify this and then test infectivity in the chimpanzee model. And of course, there was actually no animal model. Only chimpanzees were the non-human host for this infectious virus. So that left the field to really just sort of starting with sort of in the early 90s, what we knew from the sequence from Mike Houghton and his Chiron colleagues, which is shown here. Basically an RNA structure at the 5' end of the genome, which actually promotes internal initiation of translation, and then a long protein coding region of over 3,000 amino acids. But how is this sort of processed into the proteins that the virus might need in order to replicate? And Arash Grakui, who was a technician in the lab at that time, really sort of took on the challenge of, along with groups in Europe and Japan, of actually figuring out what proteins were made from this long open reading frame. And what he and others found was that there were 10 hepatitis C virus proteins, shown here, proteins involved in sort of, we believe to be the sort of virion structure, the virus particle structure shown in blue, and then non-structural proteins presumably involved in the RNA replication machinery shown in green. And this was catalyzed by a combination of host and viral proteases. And even without having sort of a functional way of testing the importance of these events, this did sort of uncover the protein boundaries and allowed people to begin to look for enzymatic activities that the virus might need that could be possible drug targets. So that included an NS3,4A complex, which is a serine protease helicase, whose structure was later determined, and also NS5B, the RNA-dependent RNA polymerase of the virus. So that's where we were, but we were without any ability to actually sort of test the importance of, say, these cleavage events on the ability of the virus to sort of replicate. So I have to digress a little bit and tell you, a little bit about what RNA virus infectious clones. I mean, why are these something that folks like me go after? So if you look at sort of a normal virus infection, you've got a virus particle that is designed to efficiently deliver this genome RNA to a cell. If you have a permissive cell type, the virus can infect that, and you get a burst of progeny viruses. At least for positive strand RNA viruses like hepatitis C, and now for SARS-CoV-2 as well, if you actually take that RNA out of the virus particle and you can introduce it into susceptible cells, it basically functions as a messenger RNA, so it can sort of set off the entire life cycle of the virus. So one of the things that RNA virologists like to do is to actually force this RNA genome through a DNA intermediate. That allows you to sort of use DNA manipulations to introduce whatever changes you want. And then you can either transfect that sort of DNA construct into cells, and if it gets transcribed into a functional RNA, the life cycle takes off, or you can design it more precisely to actually use a RNA polymerase derived from phage to produce basically unlimited quantities of basically a synthetic mimic of the viral genome RNA. So we began to try and accomplish this for hepatitis C. And the question was, you know, sort of what source of HCV RNA should we use to sort of derive these clones? And we used the sort of famous 877 serum from Harvey J from patient H. This was highly infectious. It had been titered in chimpanzees by Bob Purcell, but it was precious and in limited quantities. So trying to sort of confirm that, you know, sort of you have a functional clone, how do you do it with hepatitis C? Well, one thing would be to just transfect it into a cell culture. But as I told you, we didn't have any cell culture systems that had been proven to work for hepatitis C. So, you know, that might not work, and it didn't in this case. And then the only other possibility was basically the chimpanzee model. And by taking these RNA transcripts and injecting them intrahepatically into the liver of a chimpanzee and seeing if that would initiate infection. Now that seems like a crazy thing to do, but actually Sue Emerson and Bob Purcell's lab, again at NIH, had shown more than a decade earlier, I think, that you could use in vivo RNA transfection to launch hepatitis A in another non-human primate model, the marmoset. So it wasn't such a crazy idea. It had actually been shown to work at least for one other virus. So we tried that. It failed. So that was frustrating, but we didn't give up. So we began to worry about the actual structure of the sequence that we were working with, and also the initial sort of sequence. Might there be, you know, something missing there? And one of the reasons that we focused on the 3' non-translated region was that there were conflicting reports in the literature. The sort of initial report suggested that maybe the genome terminated with poly A, like normal cellular messenger RNAs. And another series of clones that had been produced in Japan and elsewhere suggested that there might be a poly UC tract. But we knew from our work on yellow fever that there was actually a different possibility, and that was that if you looked at the yellow fever virus 3' NTR, it didn't terminate with either one of these homopolymer tracts, but rather this rather stable stem loop structure, which we had determined some years before. So Sasha Kolokolov, a really talented sort of molecular biologist, virologist from Novosibirsk, immigrated to the U.S. and joined the lab and took this on. And he developed a fairly simple, at least on paper, 3' race protocol, which, you know, many of you will think today is just sort of standard fare. But we were challenged throughout this by the fact that we had very little HCV to work with in these human samples. And so every step in this really sort of had to be optimized in order to sort of derive clones that might have arisen from the 3' end of genome RNA. So Sasha did that, and what he found was a bit of a surprise. So this is actually the 3' non-translated region of hepatitis C. Following the stop codon of this long open reading frame, there is a region which is actually quite variable among HCV genotypes. Then there's an internal poly-U, poly-UC tract, which is actually a quite variable length. And then a sequence of about 98 bases that is extremely highly conserved among different HCV isolates. And usually these kinds of RNA elements in a positive strand RNA virus are absolutely required for replication. So simultaneously in Japan, basically the same sequence was discovered by Kunatada Shimotono's lab, who was really a major player in HCV molecular biology. And so we went on with this to say, reconstruct this clone with what we thought now was the proper 3' NTR. Try again cell culture transfection to see if it would replicate. No luck there. And then the chimp model again, and we failed there. So this was really, again, a bit of a letdown. After thought, we really sort of found the missing important piece. So at this point, you really worry about everything. We were worried, could it just be the way of administering this RNA by, just injecting naked RNA into the liver of an animal? Could there be part of the genome still missing? Was the genome RNA not infectious? Could there be an essential virion protein that was required to initiate replication, or RNA modifications that only occurred in a cell that was infected by the virus and not with the sort of synthetic steps that we were using in a test tube? We knew that this virus was very error-prone. There are an estimated 1 trillion variants being generated per day in a single chronically infected individual. So that's a lot of variation. Some of those could be lethal, and of course, in those days, the enzymes that were used for cDNA synthesis and cloning were really not as high fidelity as they are today. So a solution that we came up with, at least to try and address some of these, was to construct a consensus sequence. So Sasha assembled, again, basically a series of full-length clones, actually a library of full-length clones. And then 6 of these are shown here. And if you look at the sequences across these clones, you can see these red bars actually indicate variations from what we determined to be a consensus sequence. In other words, the majority nucleotide at each position along the HCV genome. So Sasha took that information and actually painstakingly, using restriction enzymes and ligation and some PCR steps, managed to sort of assemble a consensus clone for the H77H strain of hepatitis C. So there were still a few regions that we weren't sure about, and so one of those was the 5' end, where we'd actually seen occasional extra bases there that could be important for infectivity. And then, as I mentioned, this internal poly-U, poly-UC tract, which often showed up as variable in length, and we knew that it could actually expand and contract just in sort of typical PCR sort of cloning steps. So we incorporated some silent markers to actually sort of mark these genomes that are shown here in the sort of blue colored dots. And then sort of in the sort of red and magenta, these were, again, silent mutations that were introduced to try and mark genomes that were either had the extra nucleotides or short or sort of long poly-U tracks. So again, we were off to sort of the FDA with Steve Feinstone, who did basically all of the chimpanzee work for the studies that I've mentioned, and was involved in essentially all of our sort of HCV work in the 90s and beyond. And so we had 2 chimps to sort of test these 10 different constructs, and they were each sort of inoculated individually. This chimp 1535, we just took the transcription reactions, diluted them in PBS, and injected this in the liver at multiple sites. For chimp 1536, we used much smaller amounts of RNA, but we actually used a transfection reagent that sort of aids the transit of RNA into cells. And so this was kind of another sort of Hail Mary experiment. And miraculously enough, it worked. So in chimp 1535 in blue, you can see increasing levels of circulating HCV RNA, which we had never seen in any of the previous negative attempts. In red, you can see an increase in liver enzymes. It's associated with sort of acute inflammation. The same thing happened essentially in chimp 1536. So again, it's 2 for 2. And I think, what we could tell from that, this was, I think, definitive evidence of infection, this increasing levels of HCV RNA. The animal seroconverted is shown here in green. There were elevated liver enzymes. This was typical liver pathology for experimental infections of chimpanzees, and both of these animals initially progressed to chronic infection. So we also found out that there was no need for these 5' extra bases, and a specific length of the poly-UC tract was not required. So this, left us with a system that, could initiate sort of replication and infection in chimpanzees. And this was also very similar work was reported by Jens Bucht and Bob Purcell, also for the H strain in the chimpanzee model. And I have to, again, at this point, sort of thank Bob, because he allowed Steve to conduct essentially all of our primate experiments in his primate facility at the NIH. So where did this leave us? We felt that, this really did fulfill sort of the modern equivalent of Koch's postulates, that, we had a sequence that had been basically isolated, made synthetically, and shown that it could initiate infection and cause disease. We had an opportunity to now do more clonal infections in the chimpanzee model. That opened up studies on virus evolution, host response, and the importance of potential drug targets. For instance, we could mutate the active site residues of the viral proteases and show that those were absolutely required for infectivity. Same with the RNA-dependent RNA polymerase. And finally, we had a functional, viable HCV genome sequence that we could produce in unlimited quantities. So could we use this to sort of establish a cell culture replication system? One would hope so. But that didn't turn out to be the case, because these RNAs that were infectious in vivo in the sort of most stringent model that you could imagine, the chimpanzee model, were dead on arrival when transfected into a variety of different cell types. And this same sort of situation was being seen by a number of groups around the world. So the next breakthrough really was from Ralph Bartenschogler's lab. So Ralph is shown here with Volker Lohmann. And they produced what was called a subgenomic replicon, where they basically took out sort of the structural sort of region of the genome and replaced that with the neomycin resistance gene, put in another translation initiation site, and then left in the proteins that were believed to comprise the RNA-dependent replication machine of hepatitis C, including RNA sequences at the 5' end and the 3' end. And when they did that and electroporated this into a human hepatoma cell line, HU87 cells, and selected with the neomycin analog G418, they were able to isolate colonies that grew up that were drug resistant, that actually harbored continuously replicating HCV RNAs. So this was a major breakthrough. This was the first cell-based system supporting HCV RNA replication. It allowed sort of the evaluation of inhibitors that people had been developing through enzymatic assays against the viral protease and the polymerase, and it allowed unbiased screens to be undertaken. The only kicker was that this was initially very inefficient, and only about 1 in a million transfected cells actually initiated replication. So the sort of next generation subgenomic replicons were sort of identified by efforts from a number of different labs, including the Bartenschlager lab and ours. And basically, when we sequenced the RNA that was present in these drug-resistant colonies, it turned out that it wasn't what we started with. These actually harbored sort of mutations, which, when re-engineered back into the parental replicon genome, greatly enhanced the ability of this system to initiate replications. And some of these single amino acid substitutions could actually increase the efficiency by more than 10,000-fold. So this was a major advance in terms of the utility of the system. You could cure cells of the replicons by treating them, for instance, with interferon, and ask if these cured cells were now more highly permissive for replicon transduction. And that turned out to be the case. Several of these cell lines, including these HU87.5s, were shown to be significantly more permissive for initiation of replication of these adapted replicons. So this was really, I think, a breakthrough in terms of being able to do molecular virology. This was highly efficient. We could actually successfully transduce most of the cells. That allowed us to begin to do genetics of the functions that were involved in HCV RNA replication. There were other genotypes that replicons were designed for, with reporters to facilitate screening. And this was really a major driver of drug discovery. But was this really the secret to getting a complete replication cycle? Because, as I told you, these subgenomic replicons lacked some of the proteins that we thought were important for assembling infectious virus particles. So it was a simple step to re-engineer these back into full-length genomes that were otherwise unmodified except for these adaptive mutations. They could be electroporated into these hepatoma cells, and they replicated just fine. They produced the expected proteins everything looked great. The problem was that there was still no virus coming out. So again, another roadblock was reached. I think the next sort of major step in this story was due to Takashi Wakita, a hepatitis virus investigator in Japan. And Takashi had been studying a virus from different sort of cases of post-transfusion hepatitis in Japan, and he identified one of these that actually had a rare sort of acute fulminant sort of liver disease, which he called Japanese fulminant hepatitis 1. And when he made a subgenomic replicon from JFH1, it turned out that this could replicate with high efficiency in these hepatoma cells without adaptive mutations. And people in the field were getting suspicious that maybe these adaptive mutations that we had selected to allow persistent RNA replication in these cells were somehow deleterious for downstream steps leading to the assembly of infectious virus. And several labs, and there were 3 papers published in 2005, sort of confirmed that, basically JFH full-length RNAs or ones that had been sort of modified, chimeric ones, could actually produce virus in cell culture, and that virus was actually infectious in naive cells, and it could be transmitted to chimpanzees and also to liver-humanized mice, and then virus recovered from those sources was still infectious in cell culture. So this was really the first time that we had a complete replication system for this virus, more than 15 years after its discovery. So very, very different from sort of most human viruses that we'd worked with. So this ushered in a golden age in HCV research. I don't have time to go into this, but we could study the complete virus life cycle, began to look at virus-host interactions, the association of viral replication and assembly with membranes and lipid droplets in cells, could study sort of innate and adaptive immune responses, sort of develop neutralization assays that would be sort of useful for vaccine development. And also during this sort of time, we found that HCV and related viruses were actually not that rare and actually commonly found in other species in nature. And so now hepatitis C is really one of the best studied positive strand RNA viruses. Probably the only one that may surpass it will be SARS-CoV-2 with all of the current activity in that area. So this next picture just shows you an example of where we were. We could actually even sort of devise systems, where we could watch hepatitis C virus spread in culture. And this was, I think, very gratifying to us, basically not having a virus to work with in the laboratory for so many years. So I'd like to wrap up with a few sort of comments about treatment. So I think that HCV actually, is a virus that was, sort of ripe for coming up with a cure. We already knew that from sort of the development and progression of interferon-based therapies for treating hepatitis C, but the viral genome doesn't integrate. It's a cytoplasmic RNA virus. Successful treatment had been already shown to lead to a cure, in some patients at least a virologic cure. The virus is gone, and there was accumulating evidence that if you get rid of the virus, that you lower the risk of liver disease. So to give you just a brief history, sort of back in the sort of non-A, non-B agent days, people began treating this with type 1 interferon with basically only single-digit sort of kinds of response rates. So by 91, the sort of elimination of the virus response rate was about 16%. That was sort of improved in subsequent years by combining interferon with ribavirin or making a pegylated, more sort of stabilized version of type 1 interferon. In 2011, the first protease inhibitors were approved, telaprevir and boceprevir, in combination with the interferon-based regimen at the time. That increased the efficacy, but it also led to some terrible side effects of this combination therapy. That was improved somewhat with a couple of different protease inhibitors shown here in combination with PEG interferon and ribavirin. But in 2014, that really sort of marked the sort of end of interferon-based therapies. So we had interferon-free, direct-acting antiviral combination regimens with more than 95% cure rates. So the poster children from this are the sort of NS3 4A serine protease involved in several viral functions shown here at the bottom, sort of processing RNA replication as well as innate immune evasion. The other one turned out to be the RNA-dependent RNA polymerase, NS5B. And then the sort of third player was another non-structural proteins involved in RNA replication and virus assembly, NS5A. And these are now being used, these inhibitors against these functions are now being used in a variety of different regimens. So the virus can be eliminated in 2 or 3 months. There are several different combination therapies available. A few of them are shown here. And remarkably enough, HCV is highly variable in the world, but these new drugs cover sort of almost all of the types that are out there. So really a remarkable sort of trajectory from, basically a mystery virus in the 70s and 80s with sort of a low cure rate with type 1 interferon to this landmark discovery in 1989, the virus identified, diagnostics in place shortly thereafter. 2011, with the introduction of the first direct-acting antivirals, the 75% cure rate, to in 2015, now here we are with really these and beyond, these very effective regimens. But there are still some challenges. I don't think the story is completely over. To get to this 100% cure and eradication, which theoretically should be possible for HCV because it is an exclusively human virus, we got to figure out who's infected. And since this is often asymptomatic, there's a large segment of the world's population that doesn't even know they're infected with this virus. You also have to, if you identify somebody that's infected, you have to get them into care, get them to, sort of take the drugs and eliminate the virus. Affordable treatment, this is quite varied across the globe, but certainly some improvements could be made in terms of lowering the price and making it more widely available. With interferon-based therapies, we used to be challenged by sort of some patient groups that were just very difficult to treat, like HIV, HCV co-infected individuals. Much of this seems to have disappeared in this new area, thankfully, of direct antivirals. And then virus resistance, these cocktails of combination next-generation drugs turn out to be actually very good in terms of eliminating a huge problem with virus resistance. And then we still really don't have a vaccine. So this is something that many of us think is still going to be important, and I actually hope that we learn some lessons from all of the efforts that are being put into developing COVID-19 vaccines that may be useful for making a vaccine for hepatitis C. So I want to sum up with just a few concluding slides. This success story that, at least from a molecular virology standpoint, has been a long road, but there have been many, many people involved in this, basic scientists, clinicians, people in biotech and pharma, people running these clinical trials, and the patients themselves. So it's really been a great team effort, and it's been wonderful to be involved in this. I do want to thank my mentors and special friends, just pointing out sort of Dennis Barrett, who got me interested in a research career, and Jim and Ellen Strauss at Caltech, who were the ones that sort of introduced me to the wonders of viruses and virology. In order to sort of accomplish everything, we all depend on an incredible support staff, and some of these are shown here. And then finally, on this other word cloud is basically just a word cloud of all of our sort of HCV co-authors. And this really has been a great pleasure to work with these folks. And again, this is just a small subset of people that have had a direct association with me. We've gotten a lot of great input and advances from all of the other groups around in the world. And I'd also like to thank Peggy McDonald, shown here on the left, my sort of partner in life and in science, and two of our canine support staff. And finally, I'd just like to sort of leave you with this quote that I love from Lewis Thomas from a book of essays that he published more than 30 years ago. And he says, "In the course of scientific progress, things tend to pop up. Most of the celebrated achievements had their origins in moments of surprise in the laboratory, unplanned for, unanticipated, unpredicted. No committee convened to survey the future prospects for biomedical research could possibly have guessed at the things that lay ahead. No branch of government could have laid out detailed plans for scientific needs of the future beyond asserting that what the country needed was more fundamental knowledge about the human form and function and the agents and influences responsible for disease, and in a certain sense about nature itself. Scientific research works. It is the only way to get at the underlying mechanisms of disease and the only way to learn what to do about them. And so with that, I will stop, and thank you to those of you that managed to tune in. It's been a real pleasure to be involved in this success story.
On behalf of Karolinska Institutet, I would like to personally thank Dr. Alter, Dr. Houghton, and Dr. Rice for their outstanding lectures today. Your discovery of the hepatitis C virus is indeed a landmark achievement in the ongoing battle against viral diseases, creating hope for future generations and giving inspiration to current and future researchers around the world. We very much look forward to welcoming you to Karolinska Institutet and to our beautiful Aula Medica when circumstances allow. And to all of you who have joined us today, thank you for your attention. I wish you all a pleasant day and an inspiring Nobel week. Thank you for your attention.